Dear students, now let us discuss the enzymatic control of DNA replication, because unless we'll know the requirements for the process to occur, we will not be able to know that what and how the process is going to be completed. So the most important requirement for a biological process to occur, all of you know that these are the enzymes, because they are going to control most of the biological phenomena that are occurring inside your body. And for every specific biological process there are specified enzymes. You have already studied in chapter number 3 of enzymes that enzymes are specified for a particular process, for a particular substrate, and they always present themselves in a particular way. And enzymes need specific conditions for them to perform their functions efficiently. And enzymes, they are protein in nature. Now we will talk about the enzymes that are involved in DNA replication. Now there are 3 classes of enzymes that are basically involved in the process of DNA replication, and those enzymes are: number 1 is the DNA polymerase. The second category of enzymes that is involved in DNA replication is the Primus, and the third one are ligases. Now these are the 3 classes of enzymes that are controlling the DNA replication process in a very complex and sophisticated way, and they begin the process and catalyze the addition of nucleotides to the growing complementary strands of the DNA, or the strand that is going to be formed in accordance with the parent strand. Now the most important enzyme which is obviously present is the DNA polymerase. Now we will discuss that what do the polymerases do, what are their types. The DNA polymerase is divided into 3 basic classes: 1, 2, and 3. So there are DNA polymerase 1, DNA polymerase 2, and DNA polymerase 3, of which our point of concern is DNA polymerase 1 and DNA polymerase 3. Now the DNA polymerase 1 is a small enzyme and it plays a supporting role in the DNA replication, whereas the DNA polymerase 3, it is 10 times larger than DNA polymerase 1. And this is the main enzyme involved in the DNA replication. Now the DNA polymerase 3 is a dimer. You all know what a dimer is. Dimer is a combination of 2 small molecules, 2 monomers. They give rise to a dimer. Now DNA polymerase 3, it basically converts and connects the nucleotides that are formed inside the new complementary strand of the DNA. Their main role is to convert both of them. Their main role is to convert and connect So they convert RNA primers to DNA primers and then they join them to form the new strand. Now the speed of DNA polymerase 3 is that it can join almost 1000 nucleotides per second. And one of the very important feature of DNA polymerase is that it can add nucleotide only to a chain of nucleotides that is already paired with the parent strand. An already existing chain. So the DNA polymerase, it cannot initiate the synthesis on its own. Instead, another enzyme which we have studied is Primus. It basically constructs an RNA primer of almost 10 nucleotides. This primer will have almost 10 nucleotides complementary to the parent DNA template. Then the DNA polymerase 3, it will recognize the primer and it will add it to the new DNA molecule to construct the DNA strand. Now the RNA nucleotides in the primer, they are then replaced by the DNA nucleotides. Another feature of DNA polymerase 3 is that it can add nucleotides only to the 3 end. Only to the 3 end of the DNA strand. It means that replication will always proceed in 5 to 3 direction. Nucleotides will be added in the 3 end. Now, do you remember that at 3 end which group is present? Now let us discuss how the nucleotides are added at 5 to 3 directions in the growing DNA strand.